Can you imagine a future where we can edit our genes just as easily as we edit a document? A world where genetic disorders aren't life sentences, but problems with solutions. Welcome to the revolutionary world of gene editing technologies, such as CRISPR-Cas9, which hold the potential to rewrite the code of life. In the late 20th century, scientists discovered peculiar repeating sequences in the DNA of bacteria, sequences that would later be known as clustered regularly interspaced short palindromic repeats, or CRISPR for short. These sequences, they found, were part of a bacterial defense system against viral invaders. In the early years of the 21st century, researchers identified an enzyme, Cas9, that could be guided by RNA molecules to cut DNA at specific locations. This was the birth of the CRISPR-Cas9 system. But the story of CRISPR-Cas9 doesn't stop at bacterial defense. It quickly became clear that this system had enormous potential for gene editing in all forms of life, including humans. The ability to cut DNA at specific locations meant that researchers could now add, remove, or replace genetic material with unprecedented precision. Over the past 2 decades, scientists have refined and improved the CRISPR-Cas9 system. They've used it to create genetically modified crops, engineer malaria-resistant mosquitoes, and even remove HIV from the genomes of living animals. But perhaps the most exciting applications of CRISPR-Cas9 lie in the field of human health. Genetic disorders, caused by mutations in our DNA, affect millions of people worldwide. Some of these disorders, like cystic fibrosis or Duchenne muscular dystrophy, have no cure. But with CRISPR-Cas9, we may be able to change that. By editing the faulty genes that cause these disorders, we could potentially cure them at their source. Already we are seeing the first clinical trials of CRISPR-based therapies. In 2021, a woman with an inherited form of blindness became the first person to receive a CRISPR-based treatment in the United States. In other trials, CRISPR is being used to treat blood disorders like sickle cell disease and beta thalassemia, but it's important to temper our excitement with caution. Gene editing technologies like CRISPR-Cas9 are incredibly powerful, but they also raise ethical and safety concerns. Unintended changes to the genome could have unforeseen consequences, and the possibility of designer babies raises serious ethical questions. Despite these challenges, the potential of CRISPR-Cas9 and other gene editing technologies is undeniable. We stand at the brink of a new era in medicine, one where we can correct the genetic errors that cause disease and suffering. As we move forward, it is crucial that we navigate this new landscape with care, ensuring that these powerful tools are used responsibly and ethically for the benefit of all. So as we look to the future, remember this. The code of life is no longer a locked book. We have the key and with it, the potential to rewrite our destiny.